In this procedure, whole leaf imaging is used to assess guard cell movement in response to live bacterial cells. First, the bacteria are cultured from a glycerol stock and an inoculum is prepared. Then young healthy leaves are collected and stained with propydium iodide. The stained leaves are cut and the leaf pieces are incubated on the top of a bacterial suspension. Finally, the leaf surface is observed under the microscope and stomatal apertures can be measured. The results obtained using fluorescence microscopy show changes in the stomatal aperture in response to bacteria.
The main advantage of this method over existing methods like use of epidermal peels is that the leaf in contact with bacteria is intact, mimicking the natural conditions for bacterial penetration into the leaves.
To begin this procedure, sow Arabidopsis seeds on a 1:1:1 V/V/V mixture of growing medium, fine vermiculite and perlite. Grow the plants in a growth chamber and water as needed. The plants are ready to use in 4 to 6 weeks when they have young, fully expanded leaves prior to bolting. 2 days before starting the experiment, prepare the bacterium culture. Streak Pseudomonas syringae from glycerol stock on modified LB medium and incubate for 24 hours at 28 degrees Celsius. Use fresh culture to prepare the inoculum and always start culture plates from glycerol stocks as bacteria become less virulent.